Welcome to Pro UBC Academy, which conducts classes for CSR and GRF. Today, the topic of my talk is HIV/AIDS. HIV is one of the most dreadful infectious diseases that has rendered havoc on the human population. Since its discovery in 1982, this virus has been able to fight vigorously against our immune system, and finally it makes our immune system useless. So how does it do it, and what are the features that will determine that how pathogenic this bacteria is? So let us talk about first the features of the virus. So HIV is called as human immunodeficiency virus because it causes deficiency of immune cells inside our body. And AIDS is called as acquired immune deficiency syndrome. It means a person can be HIV positive but he will not be having AIDS. When HIV progresses through its chronic cycle, at the end of the time it takes around 1 or 2 years, or say from 2 to 10 years for a patient to show the symptoms of AIDS. Now let us go directly to the HIV structure. When you look at the human immunodeficiency virus, it belongs to the category of retroviruses. Now when you look at this genome, it's made up of single-stranded RNA. And this genome is surrounded by proteins called capsids. So this makes your viron. And then it has 4 kinds of enzymes that will help it to get integrated into the host cell. Because viruses do not replicate of themselves, they need a host to show their effect. Now it also has 4 kinds of enzymes. I will label it as R, I, P, R. R refers to the reverse transcriptase. Reverse transcriptase. Because if it needs to get integrated into the host, it needs to change its genetic material from RNA to DNA. So first enzyme required is reverse transcriptase. Second is integrase, integrase. This integrase will help the DNA called as proviral DNA, but it's converted from from RNA to cDNA, and then cDNA is changed into DNA, and now that DNA needs to get integrated into the host genome. So the enzyme required will be integrase. Now we will talk about proteases. Protease is a 3rd kind of enzyme that is present within the core of the HIV. This protease, what it will do, once the HIV starts replicating within the host cells, it provides long polypeptides, and these polypeptides need to be broken down. That's why we need protease. And the 4th kind of enzyme that will be required is RNase. RNase, mostly type H. So what will this RNase H do? Once the RNA starts to form cDNA, the RNA needs to be degraded because now we need a DNA. So this RNase will degrade the single strand of RNA and the cDNA, which will convert into proviral DNA when it acts as a template. Now the virus is in the form of DNA, and that DNA is called as proviral DNA. Now that DNA will get integrated into the host genome, and whenever a host cell gets activated for replication, all these proteins will be replicated automatically. So you can imagine what virus does. It takes over your immune system, uses the machinery of your own uses the machinery for the same metabolism which occurs in us, and produces its own proteins. In other words, it hijacks our procedures, I mean the procedure of DNA replication. Now, this being said, this single-stranded RNA, which is surrounded by capsid proteins, and within this core are 4 kinds of enzymes, RIPR, I already told you about this. Now this is surrounded by something called matrix proteins. They act like bricks surrounding this capsid. And finally, over this matrix, you will find a third layer. And that third layer is your phospholipid bilayer. The phospholipid bilayer is here. So in short, you will find that the genetic material, the core and the capsid is first coated with matrix proteins, and that matrix proteins are then covered with your phospholipid bilayer. Now there are 2 important glycoproteins that are embedded in this phospholipid bilayer. Number 1 is glycoprotein 120, and it looks like this. There is a head and then there is a tail. This tail is called glycoprotein 41. So these are the important proteins that will help the virus cell, I mean this viron, to bind to the host cell. Now let us see how does it do it. Now you might be aware that CD4 positive cells, I mean these are the kinds of cells which display this, that's called cluster differentiation 4. 3 kinds of cells display CD4. Number 1, macrophages. Number 2, dendritic cells. Number 3 are your T helper cells, or you can call them as CD4 T helper cells. So these are the main cells which display CD4, that is the cluster of differentiation 4. Now you may imagine that this CD4 is very important for eliciting an immune response against any invasion, for example invasion by E. coli or invasion by Mycobacterium tuberculosis. Similarly, these 3 cells will also start an immune response against HIV. But this CD4, HIV takes use of it. It takes a different use of this CD4. What it will do is, if you look at the outer membrane of the viron, you will find there is called glycoprotein 120, as I said earlier. What will it do is, it will go to the normal cell. This viron, now having these spikes in its phospholipid bilayer, this will be a cell, the CD4 positive cell. It will have a receptor for it, and that receptor, along with it will have a co-receptor. This receptor is called CD4, and co-receptors are of 2 kinds. Number 1, they are called as CXCR4. Number 2, they are called as CCR5. So why we need co-receptors? The first thing is this glycoprotein 120 will help the viral cell to fuse with the normal, your macrophage. So this will be done by 120. 120 makes interaction with this CD4. Now this virus being like spiked and circular in nature, another part it interacts with this, the co-repressor. So it is very necessary for the virus to get integrated. If it wants to get integrated and taken inside the cell, it has to make 2 contacts. One is GP120 with the CD4 receptor, and another interaction has to do with the co-repressor. But surprisingly, in 2008, it was seen that one of the patients who was suffering from AIDS, he got completely healed. Maybe there may be a problem, and some patients have been found that they have a mutant version of this co-repressor. When this version, the core process is mutated, the virus cannot go inside the macrophage. But in normal cases, what it will do is, now once this virus interacts, what will it do? This interaction will bring this interaction. This is your macrophage. So this neuron, it will have its capsid surrounded by its double-strand DNA. It will insert its DNA, I mean this single-strand DNA, along with the capsid. And it's got 4 kinds of enzymes, R, I, P, R. So once it goes inside, this single-stranded DNA, I mean the RNA, it gets converted into cDNA. And this cDNA finally gets converted into double-stranded DNA. And that is called as proviral DNA. Proviral DNA. And once it's inside the cell, once it forms this proviral DNA, what it does is the double-stranded DNA will now get integrated into the host genome. So this is the part where it gets integrated. This is the viral genome. So whenever the cell gets activated, I mean the macrophages, when will they get activated? They will only get activated if they face an antigen or if they face a bacteria. So when the macrophage gets activated, it will start producing its own genes, I mean its own proteins, and along with them the viral proteins will also be produced. So what actually it does is it's using the host DNA machinery to produce its DNA and then produce its proteins and then produce more viral particles. So when they replicate, the macrophage bursts out and all the this virus is replicating. Now, important thing to remember here is the macrophages activate dendritic cells, and dendritic cells, when they take this virus inside, they present the antigens on the surface. This will activate these cells. When only and only when the dendritic cells will get migrated to the nearest lymph nodes. It takes 1 day for it to go, and when it reaches the lymph nodes, this havoc will be outburst. It will start outbursting in the lymph nodes, which is the primary lymphoid organ. As such, it will destroy the macrophages, the dendritic cells, and most of them, in most cases, it will destroy the CD4 T helper cell. So it means when the immune response is not there, neither the primary immune response, I mean neither the innate immune response, and it will also destroy the acquired immune response. As such, we are susceptible for opportunistic infections. For example, infection by a fungi, infection by a yeast called Candida, infection by, say, other viruses, infection by bacteria, most commonly Mycobacterium tuberculosis. So this is overall the idea of how a virus go inside our cells, most probably the HIV. And this leads to something called as chronic inflammation. So both tuberculosis and the HIV virus, they elicit chronic inflammation. Normally when we talk about acute inflammation, it's good for us. So is inflammation good for us? Yes, it is, only when it's acute. It will help the immune response. But when it becomes chronic, our own immune response, who is our friend, the inflammation who is our friend, it becomes a foe. So I think by this time we can say the person now, when can we say that the person is having AIDS and when can we say a person is suffering from HIV? So when it all depends now on CD4 count. CD4 count. If the CD4 count gets below, if it gets below 200, if it gets, normally the CD4 count is around, say, 600 to 1200 per cubic millimeter of your blood. But what happens in case of HIV is, in its acute phase, when it's going inside our body, the cell count drops to less than 500. At this time, your immune response can still protect you from the normal bacterial infections, the fungi and parasites. But in case of Mycobacterium tuberculosis, it cannot do anything. And when the cell count starts dropping further to less than 200, you can say that the person is having AIDS. I think you got it by this time. Now, this is what happens to the person who is HIV positive. It will have two forms. It will pass through two stages. One is the acute stage and then another is the chronic stage. So if you look at it, if you see it here, you write CD4 count cells and you will see what happens to the HIV virus, virus. Initially, in the first few weeks, what will happen is the viral DNA, the HIV viruses will start increasing. On the other hand, the CD4 cells, they will start to decrease because what happens actually, the virus kills the cells, the helper cells, T helper cells. So the virus CD4 count will go down, whereas your HIV virulence will go up. And it will reach a stage where our immune response starts a counterattack. With this counterattack, what will happen is this HIV virus numbers will go less, and then when immune response cannot fight against it for so many years, it tries to balance this. But when the balance turns over to the virus, this pro-viral HIV numbers raise so high. And on the other side, what you will see that the CD4 count goes initially down in the acute phase, then it recovers in the counterattack, and finally it goes down. And when the CD4 count goes down by less than 200, you can say that the person is suffering from AIDS. So now let us see how the AIDS is diagnosed. There are two common ways. One is ELISA, E-L-I-S-A, ELISA, and another is Western blotting. These are the two ways by which we can diagnose whether a person is in HIV stage or it is in, you can say, or in the AIDS stage. And now, what are the medicines that are given to the person who is suffering from HIV? Is HIV curable? There is no cure for HIV. But as I told earlier, that a person in 2008, something drastic was observed, that out of 70 million people who have HIV, one of the person just recovered completely. So the treatment for HIV is to prolong the human life. We have something called as antiretrovirals. Antiretrovirals. These that's called antiretroviral therapy, ART. This antiretroviral therapy, it consists of drug regimen. Again, just like your tuberculosis, we need a drug regimen. One single drug is not going to help in case of your HIV infection. So this antiretroviral therapy, mostly you will have different kinds of antiviral drugs. So they are called as ARVs, antiretrovirals. Among these antiretrovirals, you will find that you will have drugs for all the 4 enzymes. Against reverse transcriptase, we have drugs. Against integrase, we have HIV-1 integrase inhibitors. Against proteases, we have protease inhibitor, for example, tiprenavir. Against RNase H. So these drugs work by inhibiting the enzymes which the virus uses to get into the host. And now, finally, what we see is if the immune system is deficient, it leads to cancer, it can lead to fungal infections, it can lead to bacterial infections, and such the person is prone to infection from every side. I think this is it from ProUBC Academy. See you next time. Please subscribe our channel and press the like button, and you can visit our institute, that's at Baghat Barzullah in Srinagar, Kashmir. And next time, hope to see you next time again. Thank you very much.